So,
Yeah, we've been complaining about swarms for like weeks, and you're like, nope, no swarms.
I put honey supers with empty foundation on there, but not all of them are drawing it out. And the ones that I opened that were drawing it out, they were just filling those cells up, even though they were only a third of the way drawn out. I'm
Yeah.
kind of worried.
Yeah, and you have to be careful with those colonies because they get honey bound so easily. Because rather than drawing out wax, they just want to put in the nectar. There's like so much coming in, they don't know what to do with it.
I know, but I don't know that I want to put another. Anyway, well, guess we'll figure it out.
Hi, everybody. Welcome. Welcome to another happy hour with Randall and Dustin and myself. Dustin's going to continue to kind of keep admitting people in. I think we still have a minute, but go ahead and let us know what you're drinking and where you're tuning in from in the chat room so we can kind of see where everybody's coming in from and what their beverage of choice is. And mine is a hard lemonade in a kid's cup because that's my life right now. How about you, Randall?
Kids cup. I'm drinking tea. I actually before quarantine started, I was kind of doing a lot of fitness stuff and decided to quit drinking beer and quit drinking. And then it started and I just haven't stopped yet. So I'm gonna keep it up till probably my birthday next month. So yeah, it's just tea for me.
Dustin, how about you? You're still at work, so probably nothing much.
Yeah, just water.
Well, I think people are still coming in, so I will just go on to the next page and remind Randall to put the email list on the chat room.
I will do it.
We, let's see, did I hit record this time? Yes. We forgot to hit record last time, so that was totally a fail on our part, and we do not have a recording from last time. So I have a reminder there to turn on my record button, and we've got that. And then next session, we'll have that recording ready, and we're going to try to kind of post them out a little bit more earlier so people get a hold of them. Okay, there's just a lot of bee work right now, so both Randall and I and Dustin, we've been kind of in and out of the yards. I don't know how much computer time you've had. You were just telling us about swarming.
Yeah, we our hives are really booming. So we got our first swarm on Friday afternoon, and luckily I think it swarmed that morning and we caught it in the afternoon, so it wasn't out for too long. And it was fun. I mean, so it's not a problem yet. Like I have a feeling I'm gonna like eat my words in a week or two if they all start swarming a lot. But it had been a long time since I caught a swarm, and I was able to take my time and just, and it was really low. So I was able to just like get down on my knees and like put my hand in it and just kind of feel around.
Play with it.
Took our time to find the queen. And once we had her, we were able to shake the branch in there, and yeah, it went really well. But now that I'm seeing that a lot of my bigger hives are getting honey plugged, I'm worried that we're gonna have a lot of swarming problems. In fact, I was out there this afternoon putting tags on all my hives because in the new apiary, there's only 20 hives and we hadn't gotten them tagged yet. And there was a lot of them just gathering on the outside around the entrance, festooning off the ledge. I'm not really sure what to do about it, but on Monday I might go on and try to give them a little bit more space.
Yeah, and it's hard, especially when you don't have supers that have drawn comb. Then it's you can't wait for them to draw out the wax. They just plug up everything they can with all that nectar coming in. Right. Sometimes we'll put in, if we have no supers or drawn combs, we'll put in a deep and just let them collect the nectar in the deep and then just extract the deep. It's kind of a
Yeah,
pain, but
I think that might be a good solution. One of our concerns is that a lot of the drawn frames that we have been used in experiments, and so we wanted all of our honey frames to be brand new equipment. So well, we'll see. That's a good idea. Maybe I'll just add it on to make sure they don't swarm, and we'll just lose that honey. Not be able to harvest it. We'll see.
yeah, we also have come across a problem too. So this picture I'm sharing is maybe a problem that other people have come across is not all of our hives at the time needed treatments in the spring. So
It's not too much honey. We'll give you that hint.
So what happens is half of our yard needs treatments, half of them don't, or half of them need supers. And when we need to put treatments on, some of them have supers and some don't. So what we end up doing is we just pull all the supers off the colonies and have them babysat by a couple colonies in the yard, treat everything, and then distribute those supers back out. And so the colonies that we chose to babysit are actually two really weak colonies. They're both queen right, but they only had like 2.5 frames of bees. Dustin tried to combine one, 2 weeks ago, and it didn't combine right, or it just didn't grow.
Yep.
And then the other one was our queen bank, which we used, to house all of our queens, but only had a couple frames of brood, and it requeened. So it was just small. So rather than combining the two colonies, because there's two queens there, we just used them to babysit all of our supers, and then all the bees in the supers will then go down boxes and kind of replenish the population.
That's smart. I've never seen that before. And so what are you treating with, and how long will these honey supers be off the treated hives?
Dustin, we're treating with OA.
Okay.
We're doing oxalic acid dribble. The lab will never. Well, the plan is right now we will never do oxalic acid vapor, not because it doesn't work or anything like that, but because we have students that we need to protect.
Just the handling.
As you can see on the left or on the right there, our boxes have holes. I know there's honey. Those are honey supers.
What's up?
But still, that goes to show we have old equipment.
Oh yes,
So,
yes. I'm having trouble with them.
You need
I
lights?
think no, I overwatered and then it got super hot and I think I baked them.
Hi,
Megan.
Megan.
Bake.
I
Oh
know.
hey. Oh shit. Sorry guys. I was just.
That's all right.
So how much of that did you hear? I thought I was muted. Oh my god.
How's it going?
Good. I was just explaining my newest plant I'm growing legally in Nebraska. So.
Everybody, Megan better.
Hi, I'm here.
And she's the president of Nebraska Beekeepers Association. I'm actually going to talk about you later today too, as one of our guest speakers.
Oh, great. Yeah, so I'm out here in my shop. I'm doing my latest batik for our demonstration. It's got a dandelion and some clover and a little bee on it, everything that the bees love.
Hold on, I'm going to spotlight your video. Show them again.
Oh, okay.
Where's Phoebe?
So right now I've got a couple layers of wax and my dye that I have applied to it. I have to let it dry, and then I go back in with more wax and cover up all the spaces I want to keep that dye color, and then I apply another another layer of wax and another layer of dye, so it just kind of builds into the end result will look something like this. So this is one that I did here. It's on fabric. with a bee on it. I like this one here.
That's beautiful.
Thank you. I've got a few of them. I had irises out in the yard. So I did a little iris.
Oh, nice.
Wait, so this is all wax and dye artwork on fabric?
Yes. Yes. So you use beeswax and paraffin wax,
right?
a combination. And then you can use RIT dye, or I went the extra step and got some really high quality porcelain dyes from the art supply store. So yeah.
Yes. So Megan's going to do her batiking demonstration for our virtual GPMB Virtual Fun Day, which I'm going to talk about today partially. So we're really excited to have her.
Where?
I'm going to mute Megan.
Thank you. I've got little ones. I've got to explain some things to them.
Thank you.
What is that process called? Batiking?
Batiking, yep.
Yeah, batik, B-A-T-I-K.
It's really cool. So we've done some of that with cochineal dyeing. So in one of our insect classes, we try to teach about the historical uses of insects. And one of those lessons is the Silk Road trade and the use of silkworms for making silk, and then the use of cochineal scale insects for the dyes. And then the beeswax was that really cool batiking aspect of the textiles. So it was really kind of a cool lesson, and a lot of people really like that. But it's fun. You dye insects. You dye an insect product with an insect dye, and then you decorate it using insect products.
Cool.
Back to you want to bring up that picture again, Judy?
Sure. Yeah.
So we treated these the other hives that needed oxalic acid. We treated those last week, and then on Monday we put all of these supers back on their normal colonies. And like Judy was saying, all the bees that were in these supers from other colonies, during the night, they moved down into the lower boxes. So this process helped boost these smaller colonies so that they're a lot stronger. It's a good little trick. You can also, if you want to strengthen a hive, if you've got a weaker hive and a stronger hive, you could just switch the hives places so that the weaker hive gets all the stronger hives foragers. I did that with 2 swarms this week. We had a stronger swarm. I wish I had a picture to share of that. It was like we had an 8-pound swarm this week. We had them down, and they already were in 2 boxes, and I needed to get them up back in line with the rest of the hives because the land managers were complaining that we have too many bees in one area. But when I got to it, it was towards the middle of the day. At the same time, we had a weaker colony. It was maybe the size of my palm of my hand. It was like an after swarm, and the queen was laying and everything, and I checked them, but I moved that smaller colony down there where the larger swarm colony was, and all of their foragers went in and really boosted that colony. And I checked them the next morning, and the box was full of bees, so that really helped them out. And then I moved them up before the day really started. So just a couple tricks to help strengthen your weaker colonies.
Questions, comments, other tricks?
Hey, it's Megan again. I had I did a similar thing to the first person who was talking about honey and putting the brood box on instead of, throwing a blank super on. So I did that yesterday to kind of reduce some swarming, but also I thought they've got 2 boxes up top. They've already they're very heavy. But I'm running low on supers. So I just thought I'd throw a brood chamber on there and let them move the honey around and save that instead of swarming. So that's my experiment, which is similar to.
Yeah, things to alleviate that congestion will definitely help with swarming. And the rains
Yeah.
don't help either. So if they're all cooped up, we've had a couple severe rains, then they just are so hot and boiling out the next day.
Yeah.
A couple of days of that and they're just done. They'll just take off. And this one we talked about last time, and I just wanted to give an update. This is that package that was queen right, but that was all that was left of that colony. So we did, again, boosted this up and just gave them 3 frames of sealed emerging brood. And we checked on them and they're queen right, and she's laying up a storm, and they've already got larvae in there. And so they're doing very well. And what we did was we actually took this out of a deep and put it into a nuke with 3 frames of brood so they can thermal regulate better. And that queen and those frames were just tucked on the side because we didn't want the other bees that were in there to just kind of overrun and take over the queen. So she was tucked in between these 2 foundation frames, and then we introduced 3 frames of brood with newly emerging bees and young bees. And then when we came back, they're already drawing out wax on these 2 frames, and they've got brood and they look queen right and doing great. But again, that would not have been so successful if I had tried that in a deep. You really kind of have to kind of put them in a nuke and get them the proper space for them to thermal regulate and get that brood going.
Do you think that they're about big enough for you to transfer into a regular size deep? Or?
Yeah. Yeah. How many frames of comb are on? Okay, so what was the question, Dustin? Sorry.
Tony's wanting to know when to take his entrance reducer out, so I'm trying to ask him how many boxes he has and how many frames of comb he has. I don't know if you guys want to talk about it or if you want me to. It's whatever. Whichever.
Yeah, describe why people use entrance reducers and when's a good time to take them off.
So the entrance reducers are, what we teach is, the entrance reducers are to minimize the space that the bees have to defend the opening to the hive. When you have a small package and you install that in a spring, if there's a larger hive nearby, they could, sense or smell that maybe you're feeding them sugar syrup, so there's resources there, and they could come over and try to rob out your colony. So you want that entrance reducer set to the smallest setting. Our rule of thumb is if you have, let's say you have 10 frame equipment, when you have about 8 combs built out, then it's time for another box. That's kind of rule number 1 that we go by. When you add a second box, you want to find a frame of capped brood down from your bottom deep and move it to the top box. And then in the bottom box, you'll shove all your frames over so that you keep your brood nest together so that you're not separating things out. I know it's warm now, but it still helps the bees out. To replace that frame, you'll just take an empty out of your second box and put on the side. But you put that you put that capped brood comb up in the center of your second box. And that helps pull the bees up into the second box. Mm-hmm. At the same time, you can turn your entrance reducer over to the next largest setting. That way that gives them more airflow. When you have 7 to 8 frames filled out in your second box, then pull the entrance reducer out for the summer. When it comes fall time and it starts to get cold, you'll put the entrance reducer back in on the smallest setting and then put a mouse guard in front of it.
Yeah, and then like Dustin said, when you're pulling that capped frame of brood out from the bottom box into the top box, if you find that you already have 2 boxes on your colony, but like Randall said, your top box is getting honey plugged, or you find that there's a lot of resources of food-wise, but not a whole lot of brood, and nothing in your supers, you're going to want to open up the brood chamber in that top box so that there's brood reaching the top of that interface between your top box and your supers. If you've got food or even a couple of inches of food, they're going to see that as a nectar ceiling or a honey wall, and they're just going to deposit downward instead of moving it upward. So sometimes we just take the frames, 3 or 4 frames in the center. It's all honey and nectar, pull that out, and then replace it with sealed brood. So that sealed brood is right against the supers. And if you don't have extra frames, sometimes it's just a partial reversal of the frames from your bottom hive, from your bottom box.
Randall and Judy, Allison is asking, she's got a second year hive that has quite a few swarm cells with eggs. Should I remove these cells or should I split the hive?
You want to go so I can drink some lemonade?
Yeah, no, I guess it depends. I mean, I guess it always depends. If you feel like there's enough, if you have enough of a bee population to split the hive, that you would have 2 still strong hives without creating a really weak hive, I think that you could split it. The other option, I guess, would be to remove them and add more space for them to grow out, right? Because if you've got swarm cells going, then maybe that's a sign that there's not enough space. So depending on how big your hives are, that would definitely inform the decision. Judy, what do you think?
Yeah, I think for a lot of new beekeepers, it comes down to how much equipment you have and what kind of equipment. Because if you split a hive and then just put it on a bunch of foundation, they're not going to do a whole lot because it's a honey flow. They're just going to honey bound what they have. But if you've got drawn combs, then if you split that hive, you know, they have time to requeen and she still has room to lay and reproduce and build that colony up. So I think it depends on what you have in terms of your equipment. Is it drawn or is it all foundation? You could cut the swarm cells out and just put supers on if you don't want to deal with splitting, you know, give them some more room and make sure they have the supers and that they're moving that honey up to the top. And that again would be if you have drawn super frames and not foundation.
I would add to make sure that you're queen right before you kill the cells, because if they've already swarmed, you don't have a queen to lay any more eggs. So make sure that you have still 1 egg per cell, so you have a queen in there laying, or you see the queen. And if you see either one of those, then you could kill those swarm cells if that's the route you're going to go. You can't just go blindly in there and kill queen cells because you don't want your colony to swarm. You need to see what your hive is doing first and understand what's going on inside of it before you make your management.
Yep, and then anytime you want to split your colonies, I always recommend that you have eggs in both sides, even if you will have, you have a queen here and a queen cell here. Just make sure both sides have eggs, just in case you accidentally smush the queen or the queen cell. They can still have larvae to draw out new ones. All right, so what other questions do we have? Okay, so while the questions are coming in, I'm gonna keep going and introduce the Great Plains Master Beekeeping Virtual Fun Day. So this is in response to a lot of our collaborating beekeeper associations having to cancel their fun day events. You know, that includes the Nebraska beekeepers, the Iowa beekeepers, the Kansas beekeepers, and our Missouri beekeepers. So these are all our partners in this regional master beekeeping training program. And it's a free program that just tries to centralize some of the resources for our region. And Randall is part of it. We have a lot of beekeepers that are a part of it. And then we're continually recruiting more educators and instructors to take part of the program. So we kind of want to put on a virtual fun day, and this is going to have research talks, management talks, field demos, honey house demos, and then maker demos. So you already met Megan, and she's going to be one of our makers demos. So she's doing a batiking value-added demo. We've got Gary Reuter, who's also our Nebraska Beekeepers Association's second Tuesday seminar speaker. This next Tuesday. But he's from Minnesota, the University of Minnesota there. We've got Randall. Hey, Randall is doing a field demonstration. You're doing alcohol washes, and what are you doing?
I'm doing varroa mite monitoring demonstrations. So alcohol washes, sugar shakes, ether rolls. And then I can also talk about bottom boards, sticky traps, like that.
Yeah. And that's all out in the field, right?
Yep. So I will. Thank you for reminding me that this is next weekend.
Yes.
Find a camera person. Yeah.
Surprise.
I was like, because what? Yeah, the 13th. That seems like it's forever away. No.
Yeah, it's next weekend. We were just trying to get everything together too. We also have the USDA research team from Baton Rouge. And so that's Mike Simone Finstrom and Kate and Frank Rankovich. And they are doing some really cool breeding efforts for this new polyline varroa resistant bee. They're also doing some cool propolis research and some varroa research. So they're going to be presenting. Jamie Bayer from Iowa's Beekeepers Association, he's going to be doing his talk on varroa oxalic acid treatment, so the vapor. So he does an excellent presentation. I think this will be a lecture one. But for the IPM for Bees, he did a field demo for Randall's program in Iowa. That was really great. Taylor Reams is going to do a presentation on varroa mite behavior. We've got Marion Ellis, who's doing his comb honey production talk. And then Corey Stevens is doing a tour of his queen rearing operation, and he's the president of the Missouri State Beekeepers Association, and he's a small scale, I think he's a small scale or maybe sideliner queen breeder rearing operation. And then Jolie is going to do a value added demonstration, right? You're doing soaps, lotions, and balms. I think she's on. And hold on, I'm getting this. Oh, there we go. Okay, we've got Bee Girl, Sarah Redlair, who's an awesome bee advocate, and she does her Bee Girl organization. She does research and conservation efforts. So she'll be presenting about bee advocacy and some of her efforts with land management and encouraging land folks to plant for pollinators. Becky is also doing a value-added product demonstration. She's going to do an advanced level demonstration. And then Priya, who is a postdoc at Ramesh's lab at Oregon State University, she is a postdoc that is studying the effects of nutrition and fungicides, and she's been doing some really cool stuff showing that fungicides can actually alter the protein and the amino acids in the pollen on the bloom, because everybody's been looking at how fungicides impacts the gut and the microbes in the gut when the bee's digesting the pollen, but she's showing that it's degrading the pollen before the bee even eats it. So that's kind of new research. I don't know if she's going to present that, but it would be really cool if she does. And then Dewey Karen is also presenting. He's got a wealth of knowledge, so we kind of gave him a speaker's choice. So we're going to find out what he's going to talk about, but he does some really cool stuff on bee management, on diagnosing your colony, hive death, necropsy, I think he calls it, necropsy on his hives. Then Brad Metz is I don't know what he is now. He's not a postdoc. He works in Dave Tarpey's lab, and he runs the Queen Disease Clinic. So he basically gets all these queens and works with the queen breeders and the rearers and assess their disease and status and health. And he's playing around with a new beekeeper's guide to honeybee pheromones and chemical signaling. So he wants to try this presentation out with us, and I'm really excited to see that. And then Jen Williams just graduated. She was a PhD student in Troy Anderson's lab, and she was looking at a chemical, they're called diamides, they're a new chemical insecticide, and she's looking at their impacts on honeybees and how they are actually relatively better and less toxic than some of the older compounds. I think I missed a whole bunch of other people, so those are just the folks that gave us pictures. Randall. We still need some pictures for, we need Randall, and so Frank is the other USDA scientist that's going to talk about amitraz resistance, because I know a lot of beekeepers are reporting problems with amitraz and it not working anymore for them, but they also have been using it for a number of years. So I think his research is looking at some of that stuff. So this is Saturday, and you can see it's like from 8 to 5. There's a mix of management, demos, research, field demos, and then Sunday, again, we have a scattering of research talks, management talks, demonstrations, and field demos. And then, oh, I think that who was not on the list and we haven't gotten a picture is Jennifer Saruda is also coming. She's in Tennessee now. She's doing photography and beekeeping. And then Ginger Reuter, who is Gary Reuter's wife, does all of the mass production of candle making for the university candle sale. So she does a lot of mass production candles, and she does some really beautiful candles. And so she's going to show us her demonstration and how she goes through that process. They have quite the custom setup that, you know, because Gary's quite the carpenter.
I see you also have Jenny Mitchell there, who's one of the big judges at the Iowa State Fair, and she also runs the insect zoo at ISU. And so I think it looks like she's going to be talking about preparing entries for competitions at state fairs.
Yeah, Jolie helped us get connected with Jenny, and I think we're really excited to get a photo of her and some more information about her talk too. But she's definitely well-versed in that world of fair entries and competition, and I think everybody should partake at some point. It's a great process. It's a fun
Jenny was a student of Marion's here at UNL, and he taught her how to judge stuff, and she is very particular. She has no problems not rewarding first place if it does not deserve it.
I believe she's our honey judge at the Kansas State Fair every year for the last 5 or 6 years.
Really?
She is. Iowa State, Kansas, and she used to do Nebraska till Judy came and took her gig.
Oh, she can totally. I had no idea. Would she like it back? I don't know. She might have to fight Dustin for it. He really likes it.
Yeah, I like going to the fair for a day and getting a free meal and getting to taste honey all day. It's great. Come home with a sugar
but it would be really good to see. If she's a strict judge, then she's the perfect person to like teach everybody how to prep fair entries. Oh, and then because we're starting to get a lot of, I guess the other person I didn't mention was Melanie Kirby. So she's the postdoc that works for Steve Shepard's group at Washington State University. And she's been abroad doing a lot of the old world semen collection for integrating genetics into our stock. And so she's been working with Steve really about, on boosting genetic diversity. And she is a local queen producer herself, coming from the beekeeping industry going into research. So kind of different from a lot of other students we see. So she's awesome, and she's going to be talking on some aspect of queens and breeding. Yeah, we tried to get a program that was going to cover everything for everybody on all levels. It was a little bit hard, so we'll see how it all pans out. Oh, I guess the last person I didn't mention was the Center of Rural Affairs. They have Cody Smith and Kirsten Bailey, who's going to present about bee policy and energy, solar energy and habitat. So they've got some cool efforts there.
Yeah, Cody Smith is based here in Ames, and we are working on projects with him to promote the establishment of prairie underneath solar panel establishments. The
So
idea,
this must be you.
what?
This must be your project then.
Tangentially, I'm helping. We're applying for funding right now. But what we're trying to do is, because normally under solar panels, they just put gravel or sometimes they'll put lawn. So the idea is that we could create some sort of pollinator habitat under there. And we're going to be doing research around the effectiveness of that. But even in Minnesota, at some of these sites, beekeepers have started keeping their bees at some of the sites where they've already established pollinator habitat under the solar panels. And then they market their honey as solar honey because technically it's coming from a solar farm. So, I mean, there's a lot of different things that could come out of it. So it's pretty exciting.
Yeah, and I heard that these a lot of these areas don't mow, right? They don't really do a whole lot of spraying or weed management, so you can plant a lot and let some of it go in undisturbed for longer periods of time.
That's part of the idea. Prairies still need management, lots of management, especially in the first 3 years. But after that, once it's established, the idea is that it would be very low maintenance. It's complicated though because you have to choose plants and the placement of plants. You don't want really tall growing plants around the panels themselves, blocking out the sun, that sort of thing. But
what about bee poop on the panels?
Yeah.
Panel could be a concern if they're landing there. And so maybe that's something that we'll try to monitor as we, it seems like we're going to get funding from the Department of Energy to do a small experiment we're doing in Iowa, Illinois, and I think Minnesota. So that's one thing that we'll look at. Bee poop on the panel.
When I was in Washington as a graduate student, there was a news article and frantic TV, whatever, about some mysterious golden shower, and it turned out to be massive bee poop. It was just a nice spring morning, and there was a huge apiary, and all the bees came out and just kind of, did their cleansing flight, and for a day or two, the town was just like, what is this mysterious golden shower, this powder? And they were trying to figure it out, and they figured it out like 3 days later. Oh, it's the bees. They all flew out, and they just like smattered a whole parking lot of cars. And it was just like, weird smattering of long trails of, bee poop. So people were just freaking out. They were like, what is this?
This going back to the event, this is a fantastic, phenomenal event. I think that people should. Hopefully, people are pretty excited about it because there's something for everyone there, and the people that are talking are all really great.
Yeah, and Marla gave a COVID, beekeeping and COVID day talk for one of our Nebraska beekeeping seminars presentations. So she's going to allow us to take the recording and also have a list of pre-recorded talks. So people who couldn't make it but wanted to contribute, a lecture, like Natalia's going to do her Bee Friendly Landscape talk, and I'll do a pesticide management talk because there wasn't enough time for on the schedule. So anytime we have guest speakers, I pull myself out and put them in, and that way we have we can look at the pre-recordings. But it's nice to have the guest speakers if they're available to answer live questions. Some of them will do a pre-recorded lecture, but then do the live chat at the end, just because they're more comfortable with that part. But it'll be a blend of live and pre-recorded, and it'll be fun. Okay, and then Dustin had some cool stuff about nematodes. You want to talk about your nematode thing this week? Oops. I'm going to stop sharing so you can start sharing.
Yeah, let me get.
So we've been playing around with nematodes, and these are entomophagic nematodes that you would use to control small hive beetle larvae or small hive beetle, but they're actually attacking the pupal stages in the soil outside the hives.
So the pictures I have is kind of how this works, how you install them, I guess we'll say. I'll say that in air quotes. We're not technically, because of the university, we're not supposed to promote any one company, but we get our stuff from Arbico Organics. They have a really nice list of beneficial nematodes for any gardening issue. They actually have a nematode list, so you pick like what kind of critter you have that you would like to biologically control, and then it pairs you up with a product you can buy. So this is the most popular species of beneficial nematode to control small hive beetles. I should say not much research has been done on beneficial nematodes. We know that in a lab, they will seek out the small hive beetle larvae in the soil and kill them. They puke bacteria that gets in their sphericals, in the small hive beetle sphericals, and then like eat them from the inside out. That's what the nematologist explained it here at UNL. So, and then once it turns to a pile of goop, which is fine by me because I hate small hive beetle, the nematodes scurry in and they suck up what they need to basically like fill up their bacteria sac, and then they move on through the soil. They do need moisture to move through the soil. They can't just dig through the dirt. They're microscopic roundworms is what they are. We have used these in the past, and they seem to, then the following season, we seem to see less adult small hive beetles in our colonies here on campus. And we've heard that from other beekeepers in the area as well. So that's kind of what we're going off of. We've had a couple of small projects where we've messed with beneficial nematodes, but I think we'd like to continue playing around with them in the future to see if we can come up with some cool research that helps add to this. Ideally, you want to apply them around your colonies in the early morning or in the evening. They will die in direct sunlight. So we're going to go through the steps. So there's nothing fancy about how they come. They come in a Walmart branded Ziploc bag in gel. It says great value on the bag. So there's roughly 5 million beneficial nematodes per package. And on their site, they say that this will treat about 10 colonies. They will arrive frozen, and you have 2 weeks to install them. So first you need to get them out, and there's a sheet that comes with your nematodes. So you follow those guidelines, but you remove the plastic bag, and you want to have a couple 5-gallon buckets of water, especially if it's not going to rain for a while. You add cold water to the baggie of the nematodes in gel, and then you shake it. And then once you're done shaking the bag, you pour the baggie with the gel and everything into a pint jar, and then put a lid on that, and then you shake it vigorously for 30 seconds. And then you pour the jar through a strainer so that you're catching all the gel, and the nematodes will fall through your strainer since they're microscopic into your 5-gallon bucket of water. And then you can just dump the gel back into the plastic bag and dispose of it. And then you want to stir the nematodes up vigorously to kind of aerate them, because they will fall to the bottom of the bucket fairly fast. Then next, you pour your water into a watering can. This is super scientific, guys. And then you take your watering can and you pour the nematode solution around your hives. Like I said, 1 package of nematodes should be enough for about 10 colonies. And if no rain is in the forecast, well, I did this Wednesday and then it rained the last 2 nights, which has been great, but it's also been in the 90s. So I hope they got down on the ground and survived. But we brought a second bucket, and then we watered a second time around all the colonies, just to try to help those nematodes move into the soil. When treating, if you have a hive infestation, you cannot apply these directly in the hive. It should be applied to the soil underneath the hive, because that's how they work. Best methods for applying them are with a sprinkler style watering can. This is directly from the information guide that you get. The best applications with a sprinkler type watering can or a hose end sprayer. If you use a spray bottle, the nematodes are microscopic, but they can still clog the mesh filters. And you want to avoid the hottest and sunniest times of the day. Directly after a rain is a good time. The nematodes will begin working for you as soon as they get settled in the soil and they get hungry. And then kind of a duh statement, but never apply chemical nematicides after applying your nematodes. And that's a little bit on beneficial nematodes, guys.
Dustin, you have a question about chlorinated water and whether that affects the nematodes.
You know, I don't know.
Yeah.
I don't know.
What was the water source you used, and was it still effective?
It was just tap water, and our water's chlorinated. And we've used that in the past and saw a drop in beetle numbers.
Yeah. I bet it's so dilute that it probably doesn't affect them.
There's nothing in here that I've got the sheet right in front of me. There's nothing in here that says that you can't use chlorinated water.
Yeah, and sometimes they'll say you only use distilled water or, you know, something that says not chlorinated. But if it doesn't say it, then I would assume that they have to guarantee the product will still work.
Yeah.
Right.
But yeah, that's
There is a time like limit on the package, right? You have to use it at a certain point, by a certain point.
yeah, you've got about 2
2
weeks
weeks.
to you got 2 weeks to put them in, and then they're not they're dead. Really it says on the package, use as soon as you get them.
When you order a package, how expensive is it, and how far does it go?
Good question.
How many hives are you able to kind of treat around with one package?
$36 for 10 hives, basically.
Okay.
We've only done the pollinator garden yard once, right? I mean, did we do it again this year?
We've done it again this year, and then we did we have hives out at Stock Seed, and we did that yard too because we had a small hive beetle slime out last year. So I did one out there, and there's only 4 hives, so I just drenched around them really good and around the area where we had the hives last year to just try to cover a good amount of space. But we did the pollinator garden two years ago, and we were doing a small experiment with the beneficial nematodes then. So we actually applied 3 treatments of beneficial nematodes in the pollinator garden. So we really doused the area with it, and that was just because the experiment that we were doing we needed fresh nematode water, so we had to buy another package, and then the experiment only required just a little bit of nematode water, so the rest we dumped in the pollinator garden. And then we've seen beetle larvae this year in the weak colony that Judy was talking about that I tried to combine. So we just thought it would be a good idea to get some in and get them in the yard.
But since we started, and this is not scientific, so we don't have any data to prove this, but since we started using the nematodes we've been using less of the Swiffer pads.
Yeah. And at least here in Nebraska, the adults seem to show up more in August, end of July and August. Since I've got to deal with it and small hive beetle are gross, like I'd rather be proactive. So I wanted to get a nematode treatment on since it seemed to work. Like Judy said, we don't have any scientific data to back it up. I think it'd be really hard to do an experiment on this because you have so many wild things in the soil.
Mm-hmm. You have a lot of species of nematodes too.
Yeah. So IDing them and all your other critters in the soil. Our pollinator garden is pretty, I would say the soil's kind of rich because we've got a lot of clovers and stuff in the yard. So, yeah, like how would you do that in the quote unquote wild? So I think that's one of the things that makes it hard for researchers to really, prove scientifically that these things work in the wild. They can do it in the lab because we can autoclave soil to where it's sterile, and then we can introduce small hive beetles into soil and then put some drips of nematode water in there and see if they die or not. But yeah.
Wow. There you go, nematodes. Anybody else have any experience with nematodes?
No, I just want to, commend you for.
Sorry, Megan, we can't hear you. Can you speak up louder? Uh-oh, I think we lost her audio. While we're waiting for her to kind of pipe back in, I did repost the link to the virtual fun day link, and there was a question about registration. So the registration form just takes down your information and your email, because we're going to send the Zoom link, the webinar link, and the access link later. And so this is a free event, but it is a charitable event, so that because of COVID, we didn't want to put any kind of payment or financial strain on anybody. So if people want to, they can donate to their association. They can donate to both their association and GPMB, or they can just donate to GPMB or none. So those will be the options. And because we're still working out how to do that with the university, it has to come out in a separate link. So we'll bring that out. We'll probably post that on Friday to give everybody who registered the access to the webinar and the donation link. And then Randall just reposted the email list so that if anybody wants to sign on to the newsletters, we can send out reminders. Randall, did you want to say anything else?
Just that I forgot to send out a reminder email this week. My bad. But yeah, and I'm happy to. How would you like me to spread the word to Iowa beekeepers about the event next weekend? Okay.
Yep, and if any of you, for those of you who have your own bee clubs and associations, please, pass this email and this Facebook event and the registration link out. It's not restricted to GPMB people. It's an open public event. Jacob, thanks. Get some Californians in there. Okay, so do we have anything else? Otherwise, we just have a couple more minutes. Any lingering questions or thoughts?
Can you hear me?
Yeah, we can hear you.
Okay, great. I just want to thank Dustin and you for doing the nematode, research. I use diatomaceous earth, but I've been pining for nematodes because we also have the Japanese beetle that comes out and just tears apart everything but tomatoes in my yard. But knowing that there are the nematodes for the small hive beetle is awesome as well.
Yeah.
Go ahead, Judy. I want to look first before I say something.
Oh, I was going to say, I've also heard other folks using chickens.
Oh,
Yeah.
got plenty of those.
They just let their chickens free roam around, and they'll kind of pick at them and pick up, pupae and larvae coming out.
They really like the adult beetles, because I'll go out and shake my raspberry bushes and just thousands of beetles will fall to the ground and my chickens are all over that.
I should take a video of that. That would be very satisfying to watch.
Yeah. I think I have heard that ducks do a better job with the Japanese beetles, but I don't like having ducks. They're too messy. They're great for the pond, but not for my my porch, which they've messed that up.
Yep, and Buzz Vance would just made a comment that nematodes feed on other insect larvae as well, so not selective for small hive beetle, and that is absolutely true. Yes, most of these are generalists. There might be some that are specific, but yeah, they will target other insect larvae.
And Megan, it looks like on their site they do have a specific beneficial nematode for Japanese beetle that you can spray.
Awesome. Yeah. I need like for the whole county, the whole. They love soybeans, and we're just, you know, surrounded by corn and soybeans out here. So whatever they can, when they spray the soybeans, then, you know, whoever's left over comes right into our garden.
Oh yeah.
Yeah.
Okay.
There's at least on their site, that's what that strain of nematode, that's what other things it feeds upon that they list.
Good. Okay.
But they have,
we had
I can copy this link, like here, you guys can see, like, this is their nematode selection chart. So on the left, it's what's your target pest, and it's A to Z. There's cabbage maggot, carrot weevils, flea beetles, all kinds of stuff. And then they just have, to the right of that, they have the kind of beneficial nematode that you can get, and then you can just click on the right over here and on which one you need to get. So
I'm wondering about the nematodes. I mean, I'm just thinking it would be easier for me if I mixed everything up at home and then traveled and applied. Is there a concern with mixing them up, like with heat in the car or anything like that?
it seems to me that their handling instructions that you get, that they're very sensitive to heat, which is why,
Okay.
like, they come with a pack of blue ice, and I keep them in there, and we go to the yard, and then I quick mix them in the yard, since they need to be mixed up anyway, otherwise they're all gonna fall to the bottom of your 5-gallon bucket. And I just do it in the yard and water quick, because knowing me, I'd put the bucket in my car, and then I'd have something else I need to do, and then my bucket's been in a hot car for 10 minutes.
Yeah.
Sounds like everybody knows that one. All right, well, there's one final question before we wrap up. Andy wanted to know if anybody has an experience with the Simple Harmony Uncapper. It's being advertised on Facebook, and I've seen it too. It's kind of a cool little uncapper. I thought about it myself, but I have not had that experience. Dustin?
It's a little spendy.
Have you used it?
Oh yeah, I seen you a picture of it right now. Yeah, yes. When the FBI does their group harvesting, their big honey party, yes, and they love it. I think Doyle bought his used, but he's got a video of him using it. So you just put the thing in, you pull it out, and then you go, and it's fast. That's why you like it. But it's like it's like corrugated tubes.
Yeah, I just pulled up a picture of it. It looks pretty cool.
Yeah. Beekeeper loves their toys, and this does look kind of fun.
But does it just slice lines on the caps?
Can you share that picture?
Do you want me? Yeah. I just opened it up on Amazon too. I can share my screen really quick.
Sorry.
Okay. $455 new.
Ooh, ouch.
Yeah, but you can see right here. It just kind of looks like it's got some tubes that then they spin, and you just slide your frame right between there. Pretty cool.
I want to say,
It looks like a giant, like, your little pick that you use, the little one with the red handle. That's what I have.
yeah,
And
yeah.
it
The capping
just
scratcher?
picks- yeah, the capping scratcher, and it just perforates the wax capping, and then you can spin it out. So if you had a rolling pin with spikes on it, I could do it.
I think, Judy, I think Brian has something similar where you run the frame on its side through something similar, and he doesn't like it because it doesn't seem to get all of the caps open.
Oh, it misses some.
Yeah, if
And-
you've got the caps that are like the really high cells and then really low cells, it might gum up.
Sure.
I mean, I don't know. I'm not knocking it. I've never used it. It looks similar to something that one of our beekeepers here has.
Yeah, if you looked around, I mean, I think you'll find, I've only seen positive testimonials on the FBI page.
I'm watching a video on it now.
It looks like the Slovenian beekeeper brush that they use to put their hives in to brush their bees off into a bucket, and then it comes back out with just the honey and no bees on it. Has brushes that spin, but this is harder apparently just for the cappings.
Yeah, and I feel like a lot of this equipment, all of it does better when you have fewer frames in the super. So if they're all drawn out just slightly out of the edge, all of those uncappers work pretty well. It's only when you're shoving 10 frames in a super and the frames are only barely being drawn out and the wood is getting chipped off, and that's when things kind of get screwed up. And yeah,
Okay.
run what, 9 or 8 frames in the super if you can, then that they draw all that wax out a little bit further. It's easier to get that cap.
Okay, from watching this video, I'll tell you why I wouldn't like it. Because you're running that frame down through it and it's just slicing your combs. Like, you're going to have all kinds of wax flakes that are going to get spun out in your extractor. And then that's going to come out and clog your sieve. Then you're going to have to sit there and clean your sieve out all the time. So instead, like Judy's saying, if you ran 9 frames in your honey super where they get nice and fat, and then you can just take a serrated bread knife and cut a chunk off and have clean, open combs to spin out, you're not going to clog your sieve. You can just extract honey and filter it and fill buckets and go.
I've got a secret. You don't need a sieve. You leave your honey sitting in a bulk tank for a couple days. Everything floats to the top, and you don't have to filter anything.
Yeah.
True.
Yeah, and with our bigger extractor, we use those baffling tanks, and that's exactly what we do. Is we just let it kind of sit up, and then it kind of goes filtering one direction and the other direction. I think Dustin does a lot of smaller extraction too.
Yeah, it depends. Like, I know for me personally at home, I do smaller extractions, so I wouldn't like this. But even here in the lab, like last year, we didn't have a lot of honey, so I was just doing it by hand with a 9-frame motorized, not our big extracting equipment. So the faster I can get it to pour through a sieve and fill a bucket, the better.
And also, Dustin is very aware of cleaning issues because we don't have a floor drain in our honey house. So,
Yes.
of course, right? Well, who needs a floor drain in the honey house? So we actually have to dismantle our entire setup, bring it outside, hose it down, and then wrap it back up and put it back in.
Yeah.
It's a nightmare.
And very heavy.
Yeah. Thanks, Dustin. All the work you do. Well, so I think we are pretty much done for today, but feel free to send us emails, send us pictures. You can reach us on Facebook or send us the email page. Let me just send you our contacts again. Here it is. Randall, I keep forgetting to put your social media tag on there.
Yeah, at Iowa State Bees on Instagram.
At Iowa
That's where I'm most active. We don't have a Facebook, so.
Okay, yeah, and put that in the chat box too, so we can kind of throw this on the thing.
Okay. Yeah, there's a really cool video of me catching a swarm.
I'm gonna have to join Instagram just so I can follow Randall.
Yeah, I thought about that as well. Not for Randall, not for you, but because of George Hansen. He only does.
What's your handle?
I just put it in the chat. It's at Iowa State Bees.
Okay, thanks.
Wait, Judy, who are you following? Who did you?
I wanted to join Instagram for George Hansen. He's the Oregon State beekeeper who does this beautiful encaustic painting.
Oh, cool.
Yeah, so if you see any of the American Bee Federation art donations and art auctions, he does this just amazing, beautiful, full-size encaustics. Next time I'll show you some pictures of some of the ones that he did. And this
Hansen.
this last January, he did an incredible painting of queen cells and bees. It was really pretty.
Yeah. Yeah, he's got a lot of really pretty stuff.
Yep, and it's all done with wax-based paints, encaustic painting. Mm-hmm.
Cool. I'm following him now.
All right, everybody. We'll see you next week. Have a great weekend.
Bye.
Thanks,
everyone.
Judy.
Bye.
Thank you.
Thanks, Dustin.
Thanks, everybody.
See you guys. Stay cool.
Oh, yeah.